First of all, I want to thank the organizers of this conference. I'm first time in Greece, first time in Trikala. It's a wonderful place, very amazing, very hospitable people here. And so thank you for this conference, for being here. And secondly, I want to apologize for my English. Sometimes my speech is faltering because I didn't have spelling practice for a long time. And the third thing that it would not be a very long lecture, only a few words about future directions of, maybe future directions of our studies devoted to UCP1 gene and protein and UCP1 gene expression. I think that practically majority of you know that UCP1 is 32 kilodalton protein that is localized in the inner mitochondrial membrane, and it is mainly expressed in the brain adipose tissue. In human, brain adipose tissue exerts its function basically during the first years of the life, but several studies have shown that it has its function in thermogenesis, in metabolism, even in adult humans. If there are some dysfunctions in brain adipose tissue function, it can lead to a weight gain around 1 or 2 kilograms per year. These data suggest that UCP1, as other specific proteins of brain adipose tissue, can play a significant role in onset of cardiometabolic disorders. 20 years ago, UCP1 was recognized as a candidate gene for development of some diseases as cardiometabolic diseases and diabetes mellitus, and some diseases related to obesity. Maybe these polymorphisms are the most known in association studies. There are some studies that confirm that these polymorphisms, basically these upstream polymorphisms in upstream regulatory region are associated with some disorders with reduced high density level cholesterol and with increased levels of low density cholesterol and increased blood pressure and some other disorders. The maybe most well-known polymorphism, majority of studies are focused on it, is this polymorphism in upstream regulatory region. Maybe it is enhancer region, maybe not, we don't know exactly. And if you can see, there are people from Argentina. They have reviewed about 40 papers devoted basically to these polymorphisms, and there are some contradictions in the data, but majority of them mention that this G allele is associated with reduced mRNA expression in brown adipose tissue, and with some kind of disorders, or with increased BMI, or with blood pressure, and or with long recovery process after some cardiovascular occasions. But why do we think so that these polymorphism play a significant role in gene expression? Maybe majority of studies that I have read, they refer to this paper. Practically 20 years ago it was published, and you see that this data, this significance, is based on data about practically 5 subjects, 5 people, with one of them was homozygous for G allele, and 4 of them were heterozygous. Maybe some other papers exist that speaks about, tells us about some molecular biological significance of these polymorphisms, but I don't know about them, maybe. Okay, we can assume that GLL is associated with decreased mRNA level in brown adipose tissue, in human brown adipose tissue, or in mice brown adipose tissue, but I have read one interesting paper, and I want to tell you more about it, that mRNA doesn't produce hot, they doesn't play a direct role in thermogenesis. This paper tells us about some experience with mice and cold acclimatization. So, first of all, they measured the levels of UCP mRNA and UCP protein in one fixed sample, and one sample of brown adipose tissue, and in equal quality of mRNA. You can see that cold exposure to cold leads to a dramatic increase in the level of UCP1, but then it becomes practically 50% higher than basic level after 1 and then 3 months of cold exposure in when mouse was exposed by 4 degrees of cold. On the one hand, maximal change in brownie deposit tissue occurs within hours in the cold, but we know that acclimatization, that mouse became more acclimated after a month. Researchers, authors of this paper, think that maybe the main thing is methodology of measurement. So you can see that maybe we mustn't every time rely on single volume of cells of RNA and measuring the level of some specific protein in them. Because it's better to measure the quantity of the level of UCP mRNA and UCP1 protein in the whole mice. So there is some data about previous figures, and there is results about whole mice. We see that dramatic augmentation of total UCP1 proteins have place, and it takes place during first weeks, practically maybe first months. And here is a figure of changing mRNA level. Maybe this data can be contradictive, so there is a little bit decrease of total mRNA level in during the first months, during the first weeks. After first maybe 2 days, it began to get lower, and UCP1 protein level became higher. But authors of this paper suggest that maybe there is no contradiction in this data. Maybe UCP1 gene and protein gene is regulated, its expression is regulated only on the transcriptional level. Maybe we should have more maybe we should have more investigations about transcriptional level of regulation, including the studies devoted to the role of this UCP. About them, we know that there is some clinical significance, that they are associated, are strongly associated with higher BMI, and maybe some other risk factors of cardiometabolic disorders. Okay, thank you for your attention.